Okay, so here we're just going to have a very short, brief introduction of our other nucleic acids, our RNA. And so here we're just going to talk about the three main classes of RNA and those RNAs that are going to be enzymes or have the function of being an enzyme. We're going to talk about the difference of the bases in our RNA as compared to DNA, their different structures, and what is an actual SNRP. All right, so basically, an RNA molecule is different from DNA in that it is going to be single-stranded and it has no helix associated with it. We have three major types: messenger RNA, ribosomal RNA, and transfer RNA. And all of these are going to be involved in making our proteins. So the nucleic acids packaged in RNA are for protein synthesis. We also have RNAs that can be enzymes, so they can catalyze reactions. We have RNAs that are going to remove internal segments, and these are known as ribozymes. We also have some that are going to be important in modifying RNA molecules and cleaving RNA molecules, such as transfer RNA. And some are going to be transferases that are going to help to form those peptide bonds in your proteins. Now, with respect to bases, we know that in DNA we have thymine, and in RNA that is replaced with uracil. And so evolutionarily, your body decided that there must be a way to differentiate between DNA, which holds all the information, and RNA, which is going to deliver information. It's going to be able to take that information from DNA and deliver it to make proteins. And so the need for making that uracil change is going to be, of course, in making those proteins. It's going to be in interacting with your ribosomes, building your actual amino acid sequence. And so uracil with adenine, again, forms two hydrogen bonds. It's the same as what we saw with thymine. Now, our structure of our messenger RNA is important. So the messenger RNA is going to be, in eukaryotes, have certain structures that are always present. So we're always going to contain a sequence that is going to convert the amino acid during translation. So we have our coding region. So this is our sequence that is converted to a protein. Oh, sorry, amino acid sequence. And this is the primary structure. Now, in eukaryotes, we have a leader and trailer sequences. So this is in eukaryotes, we have this leader sequence. Let me get this going. And we have a trailer sequence. The leader is going to start with guanosine caps. So the leader sequence is really what tells your body to add guanosine at that end. So that's the 5' end. You also have a start codon in the leader sequence. This is a trinucleotide start codon. And this is going to be our guanosine caps. And the cap is really to protect the messenger RNA from digestion by proteases. So RNA is not very stable, and if it gets out into the cytosol, we do have enzymes that will degrade it. And so your 5' cap, your guanosine cap, is there to protect it. Now your poly-A tail, or your trailer, is going to have a stop codon. And the poly-A tail can be up to 200 nucleotides long. And again, this is to protect from digestion. So the guanosine cap and poly-A tail are not complementary to the DNA. They are added after, and they are post-transcriptionally added. So our 5' cap and our 3' poly-A tail are not encoded. their post-transcription modification. Simply just meant they're added after you have transcribed the region of the DNA. Now we also have ribosomal RNAs, and so a few differences to point out. Our prokaryotes are going to have different sizes. Their ribosome is 70S. Eukaryote ribosome is 80S. Ribosome means that it's the complete ribosome molecule. They're made up of subunits, and the subunits for the prokaryotes are the 50S and 30S. Subunits for eukaryotes are 60S and 40S. And then these subunits are going to be formed from our ribosomal RNA sequences. So you're going to have, with respect to the 50S in our prokaryotes, a 5S ribosomal RNA, a 23S ribosomal RNA, and proteins. With respect to our 30S, you're going to have a 16S ribosomal RNA and proteins. And the same thing for our eukaryotes. We have 5S, 28S, we have a 5.8S, and proteins, and then an 18S. And so why is this all important? Well, prokaryotes have a different size of RNA, and so it can be a target for antibiotics, and it is a target for antibiotics. And so antibiotics that target a 50S or 30S will not have an effect on eukaryote ribosomal RNA. Now, the ribosomes are what's actually involved in our protein synthesis. So in the case of eukaryotes, that's going to be the 80S. In the case of the prokaryotes, that's going to be the 70S. Ribosomal RNA are going to have a lot of base pairing extensive loops involved in them in their structure, but the main point is the ribosome is the complete structure, and that is going to be in the cytosol. Now our transfer RNA look like little cruciforms, looks like little clover leaves, cross-shaped, a little off on a cross, but a little bit like a cross-shaped. And they are going to be important in carrying your amino acids to your ribosomes. Your transfer RNA are going to have what's called an anticodon, and this is going to pair with the codon on the messenger RNA. So we have 20 different amino acids, so we have 20 different transfer RNAs, one for each amino acid. We have an average of 80 nucleotides for your transfer RNAs that make up this clover leaf, and the structure again looks like a little clover leaf. We'll get more into the differ' important parts. What you should remember here is the anticodon is going to pair with the codon in the messenger RNA. And your 5' and 3' actually fold so that the ends are kind of close to each other, but it's really the 3' end where you're going to get the amino acid attachment. So you could have, for example, histidine attached here. And this would be at the 3' end of the cloverleaf. Now there are also other RNAs. We have some that are going to be primers for DNA replication. These primers are what are going to be used by our viruses. They're going to use our own primers to replicate their DNA. So DNA viruses will replicate in our nucleus. Also, influenza uses our primers. So influenza is an RNA virus that will go into our nucleus and use our primers to replicate their genome as well. We also have small nuclear ribonuclear proteins called SNRPs, and these are involved in splicing RNA precursors, so they have an enzyme activity associated with them. So in summary, there are 3 main classes of RNA. As we talked about, messenger RNA brings the code for a gene from the DNA. Transfer RNAs bring the amino acids, and ribosomal RNAs build. Excuse me. A ribosome has an enzyme activity, and it is an RNA. And we just talked about the structure of a messenger RNA, the structure of ribosomal RNA, and transfer RNAs.